Hi, this is my video on RNA translation. So what is translation? In biology, translation is the third step in protein synthesis, which is a part of the overall process of gene expression. In the second step, transcription produced a messenger RNA, which will now be processed by a ribosome in order to synthesize an amino acid chain, which will later fold to form an active protein. In bacteria, translation occurs in the cytoplasm and proceeds in four steps: activation, initiation, elongation, and termination. In initiation, messenger RNA binds to what is called the ribosomal subunit, and tRNA called the initiator tRNA with the amino acid methionine searches along the mRNA for the codon AUG, and once it finds it, it will bind to the site. So once this occurs, it triggers the binding of the large ribosomal subunit to this complex, which sandwiches the mRNA between the two subunits, thus forming a complete ribosomal unit and ending the step known as initiation. In the third step, elongation of the amino acid chain takes place. So another tRNA with another amino acid approaches the A site on the ribosomal unit. If it contains the correct codon sequence specified by the mRNA, it will bind to the site, but if it does not, it will be turned away. So in this case, GCU is complementary to CGA, so yes, in this case, it will bind to the A site. Now once this occurs, a reaction catalyzed by the enzyme peptidyltransferase will cause the neighboring amino acids to bond together, thus freeing up the first tRNA, and this will cause the ribosomal unit to undergo a change in conformation in which the A site will be exposed once again to the next tRNA, which will bind if it contains the correct codon sequence. So here we see that AGC is complementary to UCG, so in this case it will bind to the A site once again. And once again, we will see the reaction in which the amino acids from the P site tRNA will be binded to the amino acid on the A site and forming a longer chain. That's why it's called the elongation step. And once again, the ribosomal unit will undergo a change in conformation, and it will move to expose the A site once again. Now, the elongation step is terminated in the termination step when what is called the release factor binds to the A site when the codon UAG is exposed. All hell breaks loose and the ribosomal units break apart. Everything falls apart except for the peptide chain, which will now undergo a change in conformation to form the active protein it was destined to be. This is how a cell produces proteins, and that's pretty much it. So thank you for listening.